Hello, I'm Sarah Jeon, a postdoctoral researcher in Sang-Woo Kim's lab, and today I'm excited to share with you our recently published paper in Advanced Materials Technologies titled Ultrasound-Driven Highly Stable Implantable Triboelectric Nanogenerator with Human Tissue Acoustic Impedance-Matched Polyether Ether Ketone. In this study, we developed a novel and reliable implantable ultrasound-driven triboelectric nanogenerator, TENG, using PEK, a biocompatible material that serves as both an acoustic transmitting encapsulation layer and a high-performance positive triboelectric membrane. The TENG is designed to generate electricity stably inside the human body from external ultrasound waves, and we demonstrated its application for nerve stimulation. This approach offers a battery-free solution, addressing the challenges of replacing batteries in implanted medical devices, often requiring invasive surgeries. Instead, our device is powered externally, making long-term treatments far more feasible and comfortable for patients. Now, let's dive into the details, figure by figure. Figure 1 shows the design of the TENG. At the core is polyether ether ketone, PEK, used for both the triboelectric and encapsulation layers. We chose PEK for its biocompatibility, mechanical strength, and acoustic impedance that closely matches human tissues. This allows ultrasound waves to pass through the body with minimal energy loss. PEK transmits 99.94% of the ultrasound energy, whereas materials like titanium reflect much of it, reducing power generation efficiency. PEK acts as the positive triboelectric layer, while PFA, perfluoroalkoxyalkane, with AU deposition serves as the negative triboelectric electrode. When the two materials come into contact and separate due to ultrasound vibrations, they generate electricity through the triboelectric effect. This combination makes the TENG highly effective as an implantable power source. The device also shows promising results for nerve stimulation, successfully inducing limb movement in animal models, and demonstrates remarkable long-term stability over extended periods without degradation. In Figure 2, we explore the triboelectric properties in more detail. First, we performed contact potential difference (CPD) measurements using Kelvin probe force microscopy (KPFM) to study how triboelectrification occurs between PEK and PFA. After friction, we observed a 165 MeV increase in the CPD of PEK, confirming that its surface became positively charged after contact with PFA. We then used density functional theory, DFT-based simulations, to model the electron transfer process between PEK and PFA. As their intermolecular distance decreases, electrons transfer from PEK to PFA. The oxygen groups in PEK and the carbon dangling bonds, CDB, in PFA, were identified as critical contributors to this process. These findings confirmed the efficiency of PEK as a positive triboelectric layer, supporting its use in our device. Figure 3 highlights the electrical performance of the TENG when immersed in water, which mimics the acoustic properties of human tissues. In our finite element method simulations, we found that 20 kHz ultrasound waves cause small displacements in the PEK membrane, enough to close the air gap within the device, triggering contact separation between the PEK and PFA layers. This motion generates electricity via the triboelectric effect and electrostatic induction. In the experimental setup, the device produced 11.5 V and 52 UA when exposed to ultrasound in water. Impressively, the TENG demonstrated long-term stability, maintaining a steady output for 5 continuous hours, proving its reliability as a power source for medical applications. In Figure 4, we tested the TENG in vivo by implanting it in a mouse. We first performed an MTT assay to confirm the biocompatibility of the materials. After implantation, the device generated 8.755 V, slightly lower than in water due to the layers of tissue, but still highly effective. To demonstrate its nerve stimulation capability, we connected the TENG to the sciatic nerve and operated the device externally in water to eliminate the ultrasound's effect on nerve activation. We clearly observed limb movement during stimulation, confirming the device's potential in clinical applications. You can view a video of this limb movement in the supporting materials of our article. We also conducted ex vivo testing in porcine tissue, which more closely resembles the anatomy of human tissue. The device produced a voltage of 9.4 V, confirming its effectiveness in larger, more complex biological environments. In summary, our research demonstrates that the IU-TENG is a highly effective and reliable solution for generating sustainable energy within the body, especially for applications like nerve stimulation. By harnessing ultrasound and innovative materials like PEK, we've developed an implantable device that offers continuous battery-free power without the need for repeated surgeries. Thank you for watching. Be sure to check out the full paper linked below for more detailed insights, and don't forget to like and subscribe for updates on more exciting scientific breakthroughs.